Hi, everyone. Excuse me. Thank you for joining us today, this afternoon, or evening, morning, depending on what time zone you're in. Relatively small group registered for this webinar, so happy to make it more interactive. So feel free to interrupt with questions, either unmuting yourself if you'd like, or in the chat box. Feel free to just pause at any point. And so really what we want to do with this webinar is show you how one can do Brooker timsTOF analysis data using the fourth dimension that Brooker provides for both DDA, DIA, passive, label-free quantification, TMT, and if we get a chance, I may be able to show some SILAC data depending on how far we get. Let me go to the next slide. Here we go. So really what I'm hoping to show is to give an overview of what type of analysis can be performed in Xpro. I can see from the list of names, I recognize a lot. So some of you guys have been here for previous webinars, so I'll try not to repeat too much. Show some of the new features in Xpro and give a live demonstration. So if we think about the timsTOF instrument, and I'm just trying to, I'm hoping you can all show my screen, by the way. If you can't, please do let me know. And so as you can see, the way that the timsTOF is built is that you have your standard ion interface where ions get electrosprayed into the mass spectrometer, and at the front you have your TIMS analyzer, followed by an ion transfer multipole, the quad, some sort of your collision cell where your fragmentation is taking place, and then your TOF analyzer. And so what's happening is as ions are injected across an elution time, there's two TIMS analyzers that are essentially putting these ions into different packets based on their ion mobility. And these are then sequentially eluted in the millisecond range into the quadrupole. And so the MS1 is taken here. And so you can kind of see that in this visualization where each ion packet's MS is observed. And then essentially if we're doing MS-MS, we can have a second TIMS solution or passive solution creating the MS-MS spectra. And so most people here know how this works, but just as a high level, very high level, non-specific overview, wanted to show that. And so as depending on which platform you may be using, PEAKS Studio or PEAKS Online, a lot of effort has gone into building these on the same foundation and the same algorithm. And so really what's going on is PEAKS Online has been really developed to take over these large scale projects, which is especially true in the case of timsTOF data because the files are so large. And so PEAKS Studio is your traditional studio, and I'll focus mainly the talk today on studio instead of online. I've done a talk on online already and showed some of that timsTOF data, but certainly if you are an online user and want to know more about it, reach out to me. But PEAKS Studio does allow you to visualize it slightly better. The advantages to PEAKS Online is that it's linearly scalable and it's faster to analyze the data. And one of the nice things, or at least the features that I really like, is that what it allows you to do is it allows you to add files on the fly as you're going. So you can change, or you can add a new file or remove a file, and really the whole analysis doesn't have to take place again. Only the latest or whatever you're changing has to take place. And so in what I just kind of wanted to show is a sort of a brief chart of your options for timsTOF type of analysis in PEAKS Xpro. And so on the data dependent acquisition side of things, you can pretty much do everything that you would normally do either in PEAKS Online or PEAKS Studio. So we have functionality for both de novo sequencing and de novo assisted database search. And of course, subsequent to that, you can do label-free quantification, labeled quantification, so SILAC, iTrack, TMT, whichever you choose, and both have full ion mobility integration. On the DIA side of things, PEAKS Studio still allows you to de novo sequence and subsequently search DIA data. In online, that's been removed in favor of the more common library search. And of course, in Studio, you could also do a library search. And I will show you the library viewer that's newly built into Studio, which allows for the editing and manipulation of PEAKS libraries and third-party libraries as well. You can do DIA label-free quantification. We include the full ion mobility and include that into the quantification, and I hope to show you that in the demo. And labeled quantitation is not really done yet by DIA, although we are working on sort of a SILAC labeled quant potentially for future releases. And so if you've seen the webinars before, you've seen the slide. It seems to appear on every talk, but I think it's really important. And so really what it's showing is what's happening in the background when you pull in your data into any version of PEAKS. And so really PEAKS is feature-based, and it's been feature-based for the last few revisions. And so what's happening when you pull your data in is we are essentially searching for a retention time versus M/Z graph, and in that we're searching for the isotopic or standard isotopic distribution of carbon that you would expect to find in a peptide, regardless of what enzyme you digested it with. And so specifically with timsTOF and the fourth dimension, we now integrate that into this feature detection. And that's really where it pays dividends for label-free quantification and DDA for SILAC type quantification. But so essentially we're looking for this feature in both the ion mobility versus M/Z dimension, as well as the LC versus M/Z dimension to generate that profile. And what this allows us to do, and where we've improved quite a bit, is actually in the feature detection, because we're able to do a better job at detecting these features, we're able to do a better job at detecting overlapping features and separating these out and deconvoluting them into individual peptides, as well as identifying potential chimera scans in there. And so specifically for what's new in the new version, I'm not sure if anyone's using it or not using it. But again, we've harmonized the functionalities between Studio and online. And for the most part, have put in the same functionality in both deployments. And that includes de novo DB, library search, PTM Spider, TMT SILAC, and LFQ. And I won't show the data, but I can offline or separate as we have very consistent results across the two platforms. And so what's new in Xpro, well, I've highlighted this, but primarily built into the feature detection is better support for timsTOF, eye mobility, and subsequent to that is improved label-free quantification. We've also worked a lot on the ID transfer or match between run algorithm to drastically reduce the missing values. And we've put in a lot of work into the spectral library algorithm for DIA, specifically for eye mobility DIA. So depending on the instrument you're using, FAME, swath, and DIA passive. The library viewer I'll show you. And as I mentioned, the label-free and SILAC quantification has improved dramatically in terms of accuracy and sensitivity. So what I hope is to show is a couple of projects that I've run in studio. And if you're an online user and want me to show them in online, I have them in online, so I can do that as well. Feel free to just unmute and let me know. But the first data set comes from Jürgen Cox's lab, and it's a data set that they use to benchmark max quant. And think this is actually quite a good data set because it's timsTOF data with 2 samples, each run in triplicate. So there is some statistics that we can do there. And essentially it's different amounts of yeast and E. coli proteins spiked into a human one-to-one background. And so typically, I think looking, I won't go through the basics on how to set up a search. I think most people looking through the participant names here know how to do that. If not, please do reach out. The one thing that I will highlight is when you pull in a new project in your project wizard, please ensure that these settings are proper. And so, the enzyme can be overruled later, but the instrument, despite what the results look, it does impact the score in the algorithms used. So if you are using timsTOF, select timsTOF. Fragmentation for that would be CID. But critically important is the type of data. And we know that if you run DIA data with the DIA acquisition algorithm here, it will fail. And so just please be conscious of that. And if all your data is the same and you have multiple samples, you can always click this and click copy to whole project. And any of the settings in these 4 columns will just copy to the entire project. So you certainly don't have to do that manually. The other thing I will mention is within the same project, please don't include DIA and DDA data together. They're best kept as individual projects. And so this data set is, as I said, 2 samples with 3 biological replicates of each. And so what I've done is, because these are not fractions, these are actually distinct samples, I've loaded them in as distinct samples. So we have sample 1, 2, 3, 4, 5, 6. Sample 1, 2, and 3 are this condition A, and sample 4, 5, and 6 are condition B. And this is publicly available data, and I'm happy to send you the project if you're interested in it. And so basically what we did is ran a standard database search against a UniProt database with the 3 species, and what you get is your output as your summary page. And so new to PEAKS Pro, you have some more controls when it comes to the data view. And I will mention, just as a reminder, that anything that you set in the filters on the summary page corresponds to all the subsequent pages. And so whatever parameters we set here will correspond to both the exports as well as the protein and the peptide tabs and so on. And so to set FDRs, we're now using Peptide FDR in Studio, and online you still have the choice between PSM or peptide. But simply you can click FDR and you can pick your desired FDR, or if you have a particular point along the curve that you really want to pick, you can right click and copy that threshold. So at the peptide level, if we go to 1% FDR, that score will there. If we want to go more stringent, we can go down to 0.1, and so on. And then the other thing that's new in Xpro is you can now filter based on protein FDR as well. Yeah, I see your question. So here it's 25 and 0.05. I didn't. This is one of the earlier projects. For typical timsTOF, I use 20 ppm on the fragment and 0.02 on the sorry, 20 ppm on the parent and 0.02 on the fragment. Are my general timsTOF parameters. And so with the FDR, you can click 1% or whatever percentage, 0.1% if you prefer. At the protein level, the other thing you can do is if you don't like those drop-down lists, you can type in whatever you want. Or you can continue to go by 10 log p-square. And so when you do that, you hit apply, and that will subsequently affect all your data. And so when you see the summary page, if you scroll down, so you have your FDR curve, you have your peptide score, and then what you get is basically a summary table of everything that was identified total across all your samples, and then as individual sample replicates. And what's new to the summary page in Xpro is we've put in this sort of overlapping grid. If you have less than 3 samples, they show up as Venn diagrams. But essentially what this is showing you is at the all proteins, top proteins, or peptide level, the number of the sample overlap. So if we compare sample 1 and 2, which should be identical or very, technical replicates of each other, at the top protein level, there's 7,000 proteins that overlap, and in sample 1 there's 128 unique, and so that's kind of a nice way to quickly compare your samples. And then most of the graphs that you see further down are just standard. You get your M/Z distribution, you get your feature abundance, and if you scroll down a bit, you also get your software mass error. And so this is why I'm a bit wider on this one, Priya, is because I know that the machine was calibrated slightly off, and that the data was off. To answer your second question, you can go down to 10 or 15 ppm and 0.02 Daltons, and certainly it is enough. It's high enough accuracy to do de novo with. But I am a bit wider here because I do know that the cal of the published data set is off by about centering around -9 ppm in error. So I wanted to widen it out a bit so I could get sort of the shoulders of the error as well. But we do correct that in the software. And so, what does that look like when you look at the protein level? Again, just like any other data, and if you're a studio user, you get your typical data set that you can see. And of course, each of these peptides can be clicked on and interrogated based on what the spectra look like. And if there are multiple features, let's see if I can find one with like this one. But here there are 12 features that all correspond to this peptide. You can of course drop that down and view any of those features that sequence out to that peptide. Now, if you want to interrogate your peptides a bit more, which I always encourage people to do, if there's a particular peptide you want, or it doesn't, you can either right-click on the peptide and click Show in Table, and it will pull you into the peptide table. And then from here, you can actually view it. So you can right-click and see Show Peptide Detail, and it will pull you into now that peptide in the Peptide tab. And so this peptide isn't a great one. It actually doesn't have any features, which means there was an MS2 done, but there was no feature detected. So it's probably a very, very low abundant peptide. If we pick something where there are features, for instance, here, you can see that here, we can zoom in, that this is the MS2, and this is purple area is the feature at the LC-MS level, sorry. And you can make this a 3D view if you want, and zoom in if you want a nicer picture. But what's interesting is if you click on this IMMS button, we'll give it a second, just because it is gigabytes of data. What happens is it will basically load the ion mobility feature in that dimension. Let me see if I can make this a bit bigger for you. And so again, this is now the ion mobility dimension. This is the LC dimension. Here's your spectra, and here's your fragment match table. And I'm actually showing everything right now, but if we reset the default, we got a better looking table, which corresponds to the sequence. And so you can flip through if you wanted to see how this looks in different samples. You can of course pick that sample, and everything will automatically load your precursor profile, and again your ion mobility. Now, perhaps more interesting as I close this, an experiment like this is actually going to be the label-free quantitation. And so in the label-free oh, I apologize, I forgot to mention one thing. On the database search, if you click on the export button on the summary page, you can of course export the data into CSV or HTML, as well as OpenMS library and PEAKS libraries. And that's important because I'll show you why. But if we look at the label-free quantitation from this data, what we're showing here is basically everything. And so when a label-free quantitation opens, the defaults are quite high, with a significance of 20 and a fold change of 2, the bolded ones. And that's because we really want to right away highlight what's different across your groups. But a lot of people ask us, how do I see everything? So to do that, you just drop the filters down, and you should see everything. The other filters that I will mention to you are in the peptide filter table. And if you click edit here, again, you can specify a quality. So this is a score of how the algorithm thinks the quality of the spectra are, and the precursor is on a 0 to 100 basis. And then you have two choices that I did want to highlight: the peptide ID count and have at least X confidence samples per group. And so the peptide ID count is really saying, how many peptides do I have to have to quantitate that protein? But perhaps differently also is have at least X number of confidence samples per group. And so this is basically saying, how many MS IDs do I have to have in my group of, in this case, 3 samples, to include them in quantification. So if I say 3, that means that from each sample, I have to have the confident MS ID in order to keep that protein there. And so, you know, 1 is typical, but it depends on how stringent, you know, if these are technical, or you're looking at technical replicates that you expect to be virtually identical, maybe you want to increase that a bit. So I think that's an important filter to point out. Priya, do we lose peptides that don't have a feature? Yeah, so if there are no features, that peptide won't be quantified. But what we will do is look for it if you have it in a different sample. And so I'll show you that now. But basically you get your heat map, a volcano plot, depending on whichever base sample you've picked. and then your distribution. And so what we're looking at here is essentially a ratios. And so the 4:1, if you recall, the E. coli is 4:1 and the yeast is 1:2. And so here is our E. coli proteins, here are yeast proteins, and in the middle are human 1:1 proteins. And so that's one way of viewing. The other thing I'll highlight is this missing values and match between runs. And so this is the quote ID transfer, which is what we call it. And so in orange, as you see, essentially the missing value of quant values that are missing in each run. And then in blue is the percentage that is missing after identification transfer or after match between runs. And so, you know, this is an excellent experiment with very, very highly replicable samples. So these are, you know, better than what you would normally see in a true experiment settings. What you would normally see are the before ID transfers, probably around 20 to 30, and then the after around 10%. So slightly higher numbers, but again, depends on your experiment. These are, you know, two samples that are sort of a ground truth type of sample. So obviously, and shot in replicate, so they're going to be very similar to each other. And so when we look at the proteins, what we see are quantification, and you can filter. So for point of argument, I will filter on the E. coli proteins, and what that does is it now highlights, grays out everything that's not a E. coli protein and highlights our E. coli proteins in our volcano plot. This volcano plot is in fact interactive, so you can move in and click on a particular protein, and it will take you to that if you so desire. And so these are sorted by significance right now, but you can see on the high level overview that we have, if you hover over any of the quantitations, you can do this at the sample level, or you can do this at the group level, and you can see the ratios relative to our base sample. And so if we want to interrogate this a bit more or show how the match between runs work, again, we can pick a peptide and we can look at this peptide in detail. So if we look at the peptide list of this protein, we're seeing that essentially 6 peptides meet our filters for quantification, and of those 2 have been used. We will only use unique and generally the top 3 most abundant unique peptides. And so this peptide is really a subset of this peptide, as is this one. So we won't use those 2, as is number 6. And this one is a subset of that one. So we won't use that one. And that's why in this particular case, we're only using these 2 peptides. You can double click on this peptide. And what you get is essentially the extracted ion chromatogram from a representative sample, or you can select all, and they'll all show. And so what you see here are the 3 high-level E. coli samples and the 3 low-level E. coli samples. You can then, of course, go in a bit more and look at the actual features of where these were derived from, either at the retention time alignment level, so highlighted in gray is the retention time alignment, or you can look at the features, and of course, correspondingly, you can add in the ion mobility and look at the ion mobility. And if you'll see, what we've really spent a lot of time doing is ensuring that our ion mobility, that this dimension as well as the LC dimension all align as they should. And so if we find a peptide, let me go back to my proteins. And let's find a protein. What we can actually do is put our samples in. What I'm really looking for is a protein that doesn't do very well across all replicates, which may be very difficult to find in here. But what I'm wanted to show, or you can visualize it, is that if an MS2 wasn't necessarily done in one of these samples, this feature, including the retention time M over Z, as well as ion mobility M over Z, will all be transferred across, and that's how we get the match between run missing values. If that's clear. Let's see if I can. I don't see any off the top. It's the problem with showing a good data set. If we filter out the human proteins, you can see that we're now, as expected, hovering around 1-to-1, and same if we went to the yeast proteins. We're sitting around the opposite scale, around 1/2-to-1. And again, you know, any of these can be looked at, and if they are transferred, their feature will be taken, and if they're not, then they will be counted as a missing value, as in this particular example with this protein. And so, you know, I think that the label-free quant is really quite accurate and quite nice. And so, but what about DIA data, and how does this translate into DIA? So I'm actually going to close this just to keep things from slowing down too much. And so what we did was we actually created a library that the Brooker Demo Labs was kind enough to run for us, and these were E. coli, yeast, and human separate runs that were fractionated into 6 fractions, where we did a deep dive and a quantification, and built a library based on that. And so I won't show that project, but what I will show is I will now open up the library viewer. So this is a new thing in Xpro. You can click on library viewer, it brings up this nondescript big giant white box. If you click on open library, you can now come in and open your library. And so if I pick the right library, it'll take a second for larger libraries. But basically, you'll get this very familiar box. It's just reading in the library. And what you get is a number of different areas of information. And so one is the metadata. This is going to include your samples that you included in your library. You're going to get a drop-down list here that can show you things like your M over Z distribution, charge state distribution, peptide length distribution. You of course get your peptides, and you can scroll through any of these peptides if you want and click on it, and it's pretty responsive in terms of seeing those ions. And what's included in here is this IRT, or basically an internal IRT database, so you don't need to spike anything in. We will find that, and it's just a way of looking at and including the ion mobility and IRT dimension. And actually, you should see a CSS number as well for the data. And then you have your ion tolerance library. And so when you have that library, you can now save this library as a PEAKS format, however you want, and it will save it as a whole folder. And so really what this allows you to do is you can bring in Spectronaut libraries in text format, or OpenMS libraries in text format. We're expanding this capability. And so the library viewer is really used to view and save any libraries. And then of course, like currently you can go to the configuration and you can go to your spectral library and you can easily import that library into your software and use that library for subsequent searches. And so that's what we've essentially done. We've asked Brooker to run the same data set of same parameters. Go back with the same distribution in a DIA mode. And what we've done is search that against the spectral library. And so your spectral library viewer is going to be slightly different, or so not your spectral library viewer, but your search is going to be slightly different. But again, you see very similar results. I won't show the lib, but you basically get, instead of a database search, you get a library search. And from that, of course, you can do label free, and we see our same pattern of the 4-to-1 E. coli, the 1-to-2 yeast, and the 1-to-1 human, and the missing values. And so your missing values before ID transfer in DIA are slightly larger, but those are also fixed by the ID transfer. And so again, just like in the previous data set, you can go in and you can, I believe, you can filter. So again, if we filter for E. coli, you know, it's a slightly different distribution, but this is what the data is. The ratios are more in this sample set, closer to sort of 3-to-1 than the 4-to-1. And can we investigate that? Well, we can, of course. We can go in and pick peptides. I'm not sure why this one just does not look very good. Let's go back and we should be operating at a FDR of 1%. Apply those. So this is the downside to Studio with lots of timsTOF files is it is a little slow on the response, and that's just because each one of these files is so many gigabytes of data that it's constantly trying to talk to. I don't know if I've frozen here. I'll give it another minute. Actually, this may be a remote desktop issue. There we go. Okay, sorry about that. It's a connection to the office computer issue. And so when we look at these E. coli proteins, nothing is, that is technical difficulties. What is going on here? My apologies. I may have opened the wrong project with the wrong library. Okay, let's try this again. In the meantime, if anyone has any questions, feel free to ask and I'll try to answer them. I apologize again. So what happened here with that data set that I just showed is essentially the library wasn't configured. So it wasn't run on this computer that I'm on, and I didn't direct the software to where the the library was. And so with DIA data, what you need to do is if you're transferring data or something like that, is to just make sure that the library is imported in the software, and that the same library that was used to search the data is available for the software to reference to. What I can do while this is opening is actually show it to you in PEAKS Online. And so if we look at the same data set in PEAKS Online, we got very similar missing value percentages. And then if we look at the proteins and filter, you can look at E. coli. You can see they're all hovering around the 4 ratio, and just like in Studio, this is a interactive volcano plot, so you can pick your protein of interest, and of course then you can look at the peptides that are used for quantitation, as well as all the peptides for that particular protein. And if you double click on that peptide, it will, just like in Studio, open that peptide in the peptide view. And when you open that peptide in the peptide view, and this will be the same thing in Studio, what you get for that particular peptide is the aligned XIC for the samples are labeled. So here are the E. coli samples, the high samples. Here are the low samples. And of course, you can look at the ion match, whether that you'd like to look at just the match or the library query versus library match. The other nice thing, in what some people like to do, is this is showing the entire window. You can use these arrows to cycle through that window of the DIA that was set up on the instrument and view how the ions change. So clearly the Y6 is the largest, but that is the largest as well in the library. If we pick a different one, you can see here that most of the ions are aligning here along the center, and if you scroll through the window, you start to lose most of them and get different results in the library. And so that's what you'd expect. And so we're obviously going to pick the best one and center our precursor around there. But you can do that as well in Studio. And I will show you if the project opens. Stephen, if PEAKS X Plus use LCM data and LFQ, and let ID transfer from one or more. Yeah, so the same thing should work in Xpro, Stephen, but your data. So I don't know what version of X Plus you're using, what build, if it's 0219 or 0415. One of the major changes between X Plus release, so the 0219 and the later versions, as well as Xpro, is the inclusion of the Brooker SDK in reading the Brooker data. And so what that really means is in the older versions, in the older versions, if you looked at the spectrum of a timsTOF file, what you'd see is, instead of this ions that we're picking on the centroided data, you'd see the multiple centroided data filling the MS2 peak, kind of like profile mode. And so it cleans up the data quite a bit using the Brooker SDK. And so you especially see that in the DDA data. Where is the DDA data? If you pick any in the ion match. So in the older versions of the software, before we had that reader, these ions are essentially, you'd see all the little sticks that contribute to it in the centroiding process. And so what that did is it just created a lot of noise, which reduced quantitation and some ID accuracy. So I think you'll actually find that the IDs are a bit better, and but you'll also find that the match between runs probably works a little better in Xpro because we have better feature identification. So this is now opened. Let's hope that was the issue. Yeah, I still don't have this library here. Okay, well, I apologize for that. What you should see is what I showed you in online, with where the matches actually look quite nice, as we would expect. I must have removed the library unintentionally. I actually switched computers right before this. So again, I apologize for that. The other data I wanted to show is twofold, and one, so a lot of people have been asking for SILAC data, and I will show this in PEAKS Online because it really works the same way in both sets. But what this data is essentially a data set that was spiked in at a 3:1 heavy to light ratio. And so again, what this is, just 3 samples. They're actually 3 different injections of 10 nanograms, 20 nanograms, and 50 nanograms. And what you can see from the data is we get a nice view, decent amount of proteins identified. And when we look at those proteins, what you see in online is essentially an overview of all the different samples. But if we went to a view that's more familiar, let's take sample 3. What you see is your standard coverage map, and of course you'd see this in Studio as well, and your PTM modification of the heavy SILAC label on the C terminal R or K. And so when you went into the SILAC support that we now have, when you go in, you can view these proteins just like any other quant, and look at that particular ratio. So again, an interactive volcano plot. You can pick your protein, you can look at your peptide, and what happens now when you look at your peptide is you see both the survey scan, so you can see that this is a really low intense ion, just E to the 2, but you do get a nice MS2 scan from it. And what you can see here, where in the heavy is essentially, we'll only see the M1, but it's essentially the XIC from that heavy. You can drop this down and actually view the actual ratio of heavy to light. So in this particular case, there was no light feature detected, and you can change samples if you wanted. So it's the same thing in Studio, but essentially, and if we went down into something perhaps closer to a 1:1, if there is anything down here. These are typically sort of the one peptide, the low level IDs. So again, 1E2, really low level. And so, especially with timsTOF, I think it's important to really know your data. timsTOF has some weird collision energy settings that I know Brooker's working on in terms of if you look at the XICs, they're actually fragmenting quite early. And so you tend to see a lot of parent in the MS2s. And so I know they're working on that. We see parent here, and you can pretty much cycle through any MS2 and see a high parent level. And so sometimes, your intensities of your MS2s are gonna fluctuate. And so online and studio both offer ways to accommodate that in the filters. And so you can do that in the SILAC filters or in the label-free filters by basically requiring a certain amount of area to be present. And so if we look at a peptide feature, we could say that, I really want quality peptides, or I want to see things that are at least, 1E or maybe 5E2, something to play around with, certainly based on your particular data settings. I did want to mention, as we talked about DIA data, we do Fdr estimation with library searches, and I just wanted to briefly show how we do that. So if you take a peptide sequence, we essentially shuffle it from the known sequence to the randomized shuffled sequence. This leaves the precursor, obviously M over Z intact, as well as the charge state, but it does change the fragment ions. And so the fragment ions get shuffled together. And so what happens is essentially we have our LCMS, we detect our features. Here's our MS1. We know it should be coming along in this retention time. The MS2 go through the library search. We have our ID as well as the shuffled sequence, and we can then view these ions in terms of their alignment, in terms of what's right and what's wrong, and give that a score. And then that score is used to estimate the FDR. So there is an FDR parameter, and again, with your DIA search, you would set that FDR to both visualize your data as well as to for your quantification. So the last data set I'll show, maybe, is this by decoy score. Why is decoy score lower than target? Where do you, sorry, could you explain that a bit more? We're saying that so if you look at a precursor count against score, we're basically taking this data and putting it together to give good versus bad score, and we're using that to estimate the false discovery rate. So we're essentially trying to figure out how many of these ions actually, if we look for 388 and we see a nice intense peak, versus if we look for this precursor with 373, what is the peak? And so that intensity of the right versus the wrong is what's used. When you have triptych digestion, are all decoy sequences triptych? Yes, when you specify trypsin. So the way it's easier to explain with a figure. Let me see if I can pull up on our website, just because it's easier to show with at least it used to be here. Yeah. So on the basically what we're using is a decoy fusion, and so we take each target protein shown in blue and fuse the target to that decoy protein. So essentially we're going to have equal targets and equal decoys into the search. And then what we're looking for is the number of it's similar to the library search, we're looking for the number of real hits versus the number of decoy hits, which are the false target hits. So, but yes, it will be whatever you digest with or specify that in the search algorithm. Okay, this is taking its time to load the library search. Let's have a very small data set, TMT, to show you. This loads. What I can also do is try to open it elsewhere. So in online, what I can show you is a TMT data set, which I was going to show you in Studio, but we can also show it in online. And so this is just a single data set where yeast was spiked in 10.4.11.4.10 in a TMP 6-plex, and the human was spiked in either at tenfold or nothing. And so when we look at that data set, at the overview or any particular the individual sample, again, what we can see are individual peptides. And in both online and Studio, what you're able to see is the ion match. And again, it's a little skewed because of our giant parent ion here, 703.14.06. So there's your parent. If we zoom in, you get your nice ion match. And in fact, you can view the reporter ions and see the TMT reporter ions here. And so what that leads to is basically the extraction of these reporter ions into our intensity. And then if normalization is selected, you get a normalized intensity. Here in this case, we're not doing normalization, so they're identical. And then you get a ratio to whatever channel you select as your ratio. And so TMT is a nice way to multiplex many samples. Obviously with timsTOF, you only have the resolution to do a 6-plex. You can't go higher or use the 10 or the 11 or 16 that they're up to now. But you certainly can use 6-plexes and get really quite nice data sets from them. So if we look at the yeast proteins and apply, what you'll see is what we expect. We have high, and then we go down through the middle and then high again. And then that's pretty consistent through most of the yeast proteins. And so it's quite a nice way to look at data, and there's a big improvement here between PEAKS X, the original release, and then a patch release that we did, and PEAKS Xpro. But you can see the reporter ions nicely follow this sort of pattern that we would expect for these yeast proteins. And if we looked at the human ones, we also see this flip. So it's 10 to 1, so 1 to 0.1, which is on average what we see across those samples. And again, if you click any peptide, you can view the reporter ion. You can zoom in if you want to zoom in on the other ones that are low down. And so for TMT as well, it's nice if you have multiple samples, you can bridge them together using a common channel. And so certainly something to consider if you're looking at large scale fractionated experiments. It's a quite elegant way to do that. This is our library search that I wouldn't open before, but now, of course, that the webinar is over, is opening. And I won't spend too much time on showing it to you because we showed it online, but essentially they look the same. And I'm happy to send the project to you if you need it, and we hope to upload it as a demo project. And so with that, you know, I don't want to take up too much more of your time. I do appreciate you attending. And if you have questions or want me to go through anything, I can stay and do so, or you can always email me at any point, and we'd be happy to set up a time and do a webinar with you.